open this already. Good morning, everybody. Good morning, good morning, good morning. Happy Tuesday. Happy Tuesday. We're getting warmer and crazy weather. It's getting warmer. I had a turtleneck on yesterday, a coat and a hat. Went outside and I had to take all that off. It was hot. It's supposed to be in the 70s again today. All right, Zoom has been acting up a lot lately. I'm looking at you. You're already in, but it still got you in the waiting room. All right. Um, Please remember, we're going to start doing a few minutes, 10 minutes before each class with the progress learning. So please check progress learning. You have been assigned um, an activity of mitosis, meiosis to do. So please do that first. I'll give you 10 minutes to work on that. You all might have started it already because I think I tested it out with you all last class to see that you could access it. So let me know if you can access it. When you get finished with that, we're going to start genetics. So come back to Canvas and look at the introductory page. We have a lot of vocabulary with this. So please come back and take a few minutes to look that over. Okay. So first, progress learning. We're going to do a progress learning activity. This week we might be once a week. This week, either once because we're only meeting twice this week. Today and Thursday. So, there are a few questions there. You won't see a score right away because I have to go in and grade 2 of the questions, but please do look at those. I think yours is only 10 questions. I don't think so. I don't think so. I was watching a show the other day where they were trying to clone people, and it's a lot of science that goes into that cloning business. All right, so again, work on progress learning. We're only going to have a few minutes of that, and then we're going to come back and do genetics. We still have quite a few people missing. I'm going to check. There was a field trip this morning. I don't know if that would involve your class. Let's see. Excuse me. Yeah, it kind of does. So the people who are missing are on a field trip. Everybody who's missing is on the field trip. Oh, that's cool. It's the STEAM field trip. So, is that open? No, I got to reopen it. All right. I'm going to reopen the asexual reproduction assignment. It was closed. And so I'm going to open it till Friday because there's about 3 of you who want to work on that. Yes, ma'am. Gotcha. Oh, and the other person is not here. Oh, there's 2 of you who are here, so you can work on that one. All right, again, if you finish up with the progress learning, look at, uh, give you another few minutes to work on that. Look at the vocabulary for genetics. All right. If there's a question about any of the questions on the progress learning that cause you to scratch your head, just put it in the chat and we can talk about it. Uh, There were some videos. There were some videos I was going to show today. Um, I'm going to go ahead and show them. As half the class is missing, I'm going to not really start the unit, but we can watch the videos. And that way, those of you who are working on some makeup work, you can kind of give a listen, but still continue working.
The United States is
The week before the first day of my first year of teaching, well, over a decade ago, was an exciting time. It was also stressful because I'm a bit of a perfectionist, but a bit of a messy, unable to decorate my classroom kind of perfectionist. Actually, maybe I'm not a perfectionist. I wanted to make my classroom inviting and exciting, and I really wanted my students to walk in and think, this is amazing. I could never make it look like what I imagined up. But the most amazing thing did happen in the week to follow. I knew a colleague who had a friend who had a son who was going to college. Confusing. But anyway, that son could not take his pet snake to college with him. Some rule about dorms and snakes. My colleague asked, would you like it? They said they can give you the cage and everything. It could be a classroom pet. I didn't even have to think about it, of course. So Spike, a Texas rat snake, became a classroom pet. An amazing, popular one at that. He was friendly, well, for a snake. He'd let you hold him and not bite. That year I was the only one in the science wing with a pet snake, and I'm pretty sure there were some people that wondered, what was the point? Well, I'm big on relevance, so anytime I would teach a biology concept, I found some way to work in Spike. Predation? Well, let's talk about Spike's appetite for rats. Mitosis? Let's talk about why Spike even needs to make more cells. One day in tutorials, a student asked me, since Spike's parents were bred in captivity, did you ever see Spike's parents? Do you think Spike's parents looked like him? You can't ask a question like that and not expect an answer. It was a great question because we were getting close to our heredity unit. Heredity is about how traits are passed down from parent to offspring. We've made a playlist of our videos that focus on heredity, including reproduction, how to track inheritance and pedigrees, how to solve genetic problems in Punnett squares, and understanding different Mendelian and non-Mendelian inheritance. But you really can't delve into those and study heredity without understanding DNA, chromosomes, genes, and traits. And that's what this introductory video is going to focus on. So back to the student.
All right, pause it because this really is an introductory review, because we've done chromosomes and DNA and all of that. And a lot of what she's going to cover at the beginning is just a review of what we have already done, but you still need to understand that in order to do genetics. So we're still going to be talking about chromosomes. Oops, sorry. We're still going to be talking about chromosomes and DNA, except now we're going to focus on what a chromosome looks like and what the individual units on the chromosome are. So this of the two videos that I'm going to show, this one is more review than the other one.
Question. Spike has traits. The patterns on his body, his size, these traits are coded for in his DNA. Some of the traits he inherited can be influenced by the environment. For example, if Spike had not had the nourishment he needed, that could affect his size. DNA is not just one big code buried deep down in an organism like some treasure. Spike's DNA is in the nuclei of nearly all of his body cells. He inherited his DNA from his mother and father. I can't know for sure what Spike's parents look like, but I do know that Spike inherited his DNA from them. One fun fact, many snake species can reproduce asexually. Had that been the case for Spike, he would have inherited all of his DNA from only one parent. But it would still be DNA coding for traits. Just like Spike, your DNA codes for your traits, and your cells can't function without it. DNA determines how tall you are, what color your eyes are, what color your hair is, or even if you're at risk for certain diseases. Also, like Spike, your entire DNA code is in most of your body's cells. That's why in those crime-solving shows, which we may or may not absolutely love, a criminal can sometimes be caught by just leaving a cell from a hair follicle behind. DNA has a beautiful structure, and that structure will help you understand how inheritance works. DNA stands for deoxyribonucleic acid. It's a type of nucleic acid. If you remember from our biomolecules video, nucleic acids are a type of biomolecule. Nucleic acids are made up of building blocks called nucleotides. Nucleotides have 3 parts. One of them is a sugar called deoxyribose. One of them is a phosphate. We sometimes say DNA has a sugar-phosphate backbone. But the most important part of the nucleotide is the base, because the sequence of the bases can actually code for traits. So as far as the bases go, there are 4 types of bases in DNA. A lot of times they'll just use the letters A, T, C, G. The A is for adenine, the T is for thymine, the C is for cytosine, and the G is for guanine. These bases actually pair in a specific way, and there's a popular mnemonic that can help you remember which of them pair together. Apples in the tree? That tells you the A for apples, T for tree, because the bases A and T go together. The other verse is car in the garage, and that can help you remember the base C always goes with the base G. Regardless of whether we're talking about Spike, or a plant, or a protist, or a human like you, these are the DNA bases in living organisms. But the amount of DNA bases overall, and the sequence of those bases we mentioned, will vary among different species, and also among different individuals. But it's likely to infer that Spike has a sequence of DNA bases that is more similar to his parents than he would to, say, a rattlesnake. DNA has 2 strands, so there are nucleotides running up one side, and there's nucleotides running up the other side. The bases are what pair in the middle. The bases are held together by hydrogen bonds. The DNA is also twisted in something we call a double helix shape. Portions of DNA make up genes. We can say, for example, that this part of the DNA here makes up a gene. Genes can code for proteins. Proteins have a huge role in expressing a trait. For example, let's consider your own eye color. Human eye color is actually pretty complex. It's determined by many genes. The genes can code for proteins involved in producing the eye color pigment. But proteins coded for by genes, they can play a wide variety of roles besides just your eye color. Proteins are involved in transport, in structure, in acting as enzymes that can make all kinds of materials, in protecting the body, and so much more. We do want to mention that not all genes are used to make protein, and there are parts of DNA that are non-coding. And even though nearly all of your body cells have your entire DNA code, your body cells may only use certain portions of those genes. Genes can be turned on or turned off by a variety of mechanisms. We call that gene regulation. Check out our video on that. Now, you have a lot of DNA. When it's compacted, it can be organized into a unit called a chromosome. Very helpful when you're trying to make more cells and you need to get the DNA into those new cells. Chromosomes in your body involve DNA wrapped around protein structures. Humans have 46 chromosomes.
Okay, that's the number you're going to have to commit to memory. Humans have 46 chromosomes, 23 pairs. Okay, so we're going to be looking at chromosome maps and different sizes of chromosomes, but we're going to spend most of our time with humans. Every species can be identified under the microscope by how many chromosomes are in their nucleus. All right, so they know that they're looking at a human cell because it's got 46 chromosomes.
Nearly every body cell in your body has 46 chromosomes. Human sperm and egg cells, on the other hand, each contain 23 chromosomes. So you receive 23 chromosomes from your mother, and 23 chromosomes from your father to give you your 46 chromosomes, your genetic code. So let's do a recap of the big picture. Here's a single chromosome. You see genes on this chromosome. These genes consist of portions of DNA. DNA is made up of nucleotides, and it's these bases here, the sequence of them, that makes the difference in coding for traits. Whew. Understanding this foundation is essential for understanding heredity, whether you're talking about you or Spike. So this may bring up more questions now, like where do dominant and recessive traits come in? What about alleles, and how do you arrange those around a
Okay,
Punnett?
all of this is what our next unit is about. So we're going to be talking about dominant versus recessive. We're going to look at alleles. And again, alleles basically are the gene. And when we look at the chromosomes and we say homologous chromosomes, those are vocabulary words we had last unit. Homologous chromosomes are chromosomes that have the same kind of information on them. Like this one could be for eye color, this one, but they're both coding for eyes. And this one could be blue eyes, this one could be brown eyes. Okay, you got these letters out here are whether they're dominant or recessive. So we're going to talk about that next. And this little box over here is called a Punnett square. And we're going to have so much fun drawing Punnett squares. This is always the thing that is challenging for everybody to get done. So this is what you have to look forward to.
Square. Well, you can explore those concepts and more in our heredity playlist. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Alrighty. So that's the first one. So like I said, that one was a lot of just review of DNA and RNA and what DNA is, and some of the things that you did last unit when we were talking about protein synthesis and the making, what a DNA molecule looks like. So they talked about it, the base, the nucleotides, and all of that sort of thing. So we're not going to focus so much on nucleotides this time, because we're just going to focus on the fact that the chromosomes have segments called alleles or genes. That's what we're going to focus on. So the next one.
¿Llegamos a tiempo?
Difícil, hermano.
It's going to talk about alleles and genes.
I don't remember which grade it was where I learned something about my taste buds that can never be unlearned, but the event in the lesson with genetics has stuck with me forever. For you see, I learned that my taste buds cannot taste PTC. PTC stands for this. We'll stick with PTC. And it's a chemical that can be sold on these paper strips. It can be purchased under the name PTC paper, and it's popular in genetic classes because it has this fascinating quality. Some can put it on their tongue and immediately say, yuck, this is bitter. And some people, when they place the paper on their tongue, taste absolutely nothing. Well, unless you consider the paper. Does paper have a taste in itself? That's a debatable question, but the point is some people can taste PTC and some people cannot taste PTC. And I was really disappointed because I remember that I was the only one there that could not taste it. So there was everyone getting this amazing science experience and I couldn't taste a thing. Well, there may have been more than just me that couldn't taste it in the classroom that day, but they didn't seem as concerned by the fear of missing out of the PTC paper as I was. I remember someone trying to make me feel better, saying, "Oh, but it tastes bitter. You're actually lucky." Then they tried to describe what it tasted like to me. But it's not the same. I guess I'll never know for myself what it would have tasted like. Of course, the reason PTC paper is used in genetic classes is because the trait of being able or not being able to taste PTC is based on genetics. A reminder from our Intro to Heredity unit that genes are portions of DNA, and they have the ability to code for a characteristic, a trait, like being able to taste or not to taste PTC. Now, we do want to point out that many traits are actually coded for by interactions of more than one gene, like eye color, which is quite complex and determined by interactions of many genes together. In fact, the ability to taste PTC or not may involve some other gene interactions. There's even different ranges for how bitter the chemical may taste because there may be more kinds of alleles than we'll mention. More about that later. But since we do know that the ability to taste PTC or not taste PTC is at least heavily impacted by a specific gene, it does make it powerful for genetic classes. One thing I have found so interesting is that my parents can both taste PTC. So why can't I? Recall that humans have 46 chromosomes. Chromosomes are made up of DNA and protein. It's a condensed unit of DNA. My whole genetic code is represented by these chromosomes. You inherit 23 chromosomes from your mother and 23 chromosomes from your father. Here's all 46 of them right here. As you can see, there are 23 chromosome pairs. Each pair.
All right, I have a challenge for you. So this is what we call a chromosome map or a karyotype. So we're going to be looking at these. Can you tell whether, just by looking at this, whether this is coding for a male or a female? Is this a boy or a girl? If you could tell the difference, tell me why you know that. Is this a boy or a girl? Just type it in the chat. All right, Raymond says it's a girl. Why do you say that, Raymond? You're right. Why do you say that? It's pink. Well, that's probably what they probably did that on purpose. Yes, but that's it's a girly color. But that's not why. But that's an interest. They probably did do that because it's a girl. But can anybody else figure out why it's a girl other than the pink color? What do you know about chromosomes? What do you know about chromosomes? How do you know a boy from a girl with the chromosomes? There's only one set of chromosomes that tell you whether it's a boy or a girl. All the rest of them are body's chromosomes. Nobody knows. Ah, yes, the X. All right, the X chromosome, and there's two X chromosomes. So there's no Y chromosome. If it was a boy, one of those would be on the X and one would be on the Y. Okay, because that would be the pair. But both chromosomes are over the X. That means there's two X chromosomes and no Y. So this is a girl. Girls have XX and boys have XY. All right, so we're going to be talking about that too as we go through this unit.
Pair has one chromosome from one parent and one chromosome from my other parent. If we focus on one of these pairs of chromosomes where the PTC taste sensitivity gene may be found, we can see an area where the PTC taste sensitivity gene could be. Let's assume this is the locus, where the PTC taste sensitivity gene is found. See how it is pointing to a specific area here? That's because it's on an area of the chromosomes that refer to a specific gene that codes for a trait. Now, remember how this chromosome is from mom. This one is from dad. Each parent contributes an allele, which is a variant of a gene. An allele is a variety of a gene, a form of a gene. The alleles could be the same form of the gene or different forms of the gene, but regardless, in this case, they're forms of the gene involved with PTC taste sensitivity. So, if PTC taste sensitivity is being used as a one gene trait example, and as we mentioned, it may not be that simple, then your DNA code has a gene related to PTC taste sensitivity. Together, the two alleles you inherit, the forms of that gene, determine the trait of tasting PTC or the trait of not tasting PTC. That gene is involved with coding for taste receptors on your tongue, and the receptors you have can make a difference for whether you taste PTC or not. The alleles are typically represented by letters. Since this is all about tasting, let's use the letter T. But wait, it matters whether I represent it as a capital or lowercase letter. If I use a capital letter to represent an allele, it means that it's a dominant allele. If one or both of the alleles you inherited for a trait are dominant, then it will be expressed. More about that later. If I use a lowercase letter to represent an allele, that means it's a recessive allele. Recessive alleles are typically not expressed unless there is no dominant allele present. Now remember that you have two allele copies, so the combination you could have here could be big T, big T, big T, little t, or little t, little t. These are called genotypes, your genetic makeup. Genotypes can help determine a phenotype, which is a physical characteristic. You'll notice when writing genotypes, I put the capital letters first, if it contains a capital letter. That's not because the order matters, it's a formatting formality that capitals are written first. It turns out that being able to taste PTC is a dominant trait. That means the phenotype, which is a PTC taster, is due to a genotype that includes at least one dominant allele. So which genotypes can taste PTC then? Well, big T can. Both of those alleles are dominant. So can big T little t because remember, it only takes the presence of one dominant allele. In fact, the only genotype in this simplified example to not be able to taste PTC would be little t. So obviously that is what I am. I am the little t genotype, which results in my non-taster phenotype. But my parents can taste PTC. So what genotypes would they have to be? Well, if they were both big T, that wouldn't be possible. If one was big T and one was big T, that still wouldn't be possible. Remember, you have to get an allele, a form of a gene, from each parent. If my parents do taste PTC and I do not, then my parents have the genotype big T.
All right. Again, this is the kind of activities we're going to be doing. We're going to be looking at predicting, because all of this is probability and predicting and what are the chances are. So we're going to talk about that a lot. So what she's showing here is you can't get something from your parents that they didn't have. Okay, in this case, she cannot taste PTC, and that's because each of her parents gave one non-tasting gene to her. Okay, so they had to be carriers, and we're going to call these people carriers of the gene. All right, so we're going to be looking at genotypes and phenotypes. So the genotype is what genes you actually have. Phenotype is how is it expressed. So she's a non-taster. That's her phenotype. Her genotype is little t.
And their phenotype is PTC taster. Punnett squares can be used to determine the probabilities of offspring having certain genotypes, which then can be used to determine their phenotypes. But Punnett squares are for another Amoeba Sisters video. Before we end, one more thing to mention. In this example, the dominant trait of being able to taste PTC is more common than the recessive trait of not being able to taste PTC. And one could jump to an assumption that dominant traits are more common, especially since it only takes the presence of one dominant allele to show up in the phenotype. At least in Mendelian inheritance. But the dominant trait is not always more common in a population, because it's possible that the dominant allele itself is more rare. That can be the case with some forms of polydactyly, that is, being born with extra fingers. Some forms of polydactyly can be a dominant trait caused by the presence of at least one dominant allele. However, the dominant allele may not be as common in the population, and the condition of having extra fingers is generally rare. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, have you ever met anybody who has an extra finger? It's usually kind of like a little pinky hanging off the end of their hand. It looks kind of weird, but and they sometimes just have it taken off. So you might not know that they had one. All right, with the few minutes that are left, again, look at the vocabulary for the unit. It's a lot. And when everybody's here on Thursday, we'll go ahead and get started with this. Okay, you can get a sneak peek. See all of these lovely vocabulary words. We're going to talk about the father of genetics. We're going to talk about karyotypes and hybrid and types of inheritance, pedigree, and genetic disorders, looking at what the map is called a karyotype. So we're going to be looking at those. It's a lot to do with this unit. All right, these diseases down here to the bottom, we kind of touched on those a little bit Turner syndrome, Down syndrome, and Klinefelter syndrome. We talked about those a little bit at the end of the last unit. But again, spend some time with the vocabulary. Understanding the vocabulary is going to be important because every time we look at something, you have to tell me, is it heterozygous? Is it homozygous? Is it the phenotype? Is it the genotype? You're always going to have to be telling me this. So get to know the vocabulary. Okay, folks, if you're in a good place to stop, you can go ahead and stop and log out, and I'll see you on Thursday.